A ver. Hello. Yeah. Mira.
So much. Like you've changed so many people's and saved so many. It's just, I just.
Hi, Ana Belén. Okay, so I have muted you. I can unmute you right now. Let me see. Okay, you're unmuted.
Hi, Sophie.
Hi, sweetie. Please write hi on the chat box. Can you see me?
Yes.
Okay. Okay, cool. Thank you. Hi, Dome. You can write hi in the chat box, please.
Hi, Sophie.
Hi, sweetie. Please write hi on the chat box, and then that's how I can take your attendance. Okay. Dome. Franche. Hi, Franche. You need to write hi in the chat box so that I can take attendance.
Okay.
Okay, that's what waiting.
Hi, Sophie.
Hi, sweetie. You guys need to write hi in the chat box so I can take attendance.
What?
Write hi in the chat box. No, sorry. Okay, so I am recording. Okay. Does it tell you anywhere that I'm recording?
Mm,
Yes.
Yes. it
yes,
does. Okay, cool.
top left.
Top. Sí. Okay, people. A ver quién más acaba de llegar. Anybody that just came to the virtual lecture, please write hi in the chat box. Gary. Gary, please write hi in the chat box. And I'm thinking I'm just missing Isa. And Martin. Yeah. Okay. Okay, people. So the first thing that we're going to do, first of all, you guys can be, I'm going to unmute you. Okay. But let's not talk just yet. The reason why I'm unmuting you in a second is going to be because we're going to be taking a very short, it's just 5 multiple choice questions on AP Classroom. And to do that, you guys are going to be using that lockdown browser. Okay. Now, what I want to test, because I want to see that if this works, I want to test whether you guys can be in Zoom, like in the background. You won't be able to see me or see the Zoom web page. You're not going to be able to see that. But I want to see if you can be in the background with me so that in case...
It closes all apps automatically.
What, it closes automatically?
Yes. It says that you have to quit all the other apps in order for the lockdown browser to work, like if you have a background up.
Okay, so what we're gonna do in that case, that sucks, but in that case, what we're gonna do is I'm going to have that be the very last thing we do, so that when I let you go, you do your 5-minute,
Okay.
your 10-minute quiz, and then that way you don't have to be in Zoom with me anymore.
Hi, Sophie.
Hi, sweetie. Okay, so what I'm gonna need is, you guys are gonna have this lockdown browser, which means you won't be able to access anything in your computer, okay? But if there's any, there shouldn't be, okay? So there, you really shouldn't need me. But if there's any question that is more about technical stuff, okay, content related question, you shouldn't need me. But if there's any questions about like technical stuff, then write to the WhatsApp group. But again, that kind of defeats the purpose because I don't want you to be using your phone or using the computer. That's why there's a lockdown browser. So it's only if there's an emergency that you can write down in the WhatsApp group, okay? Okay, so we're gonna start off. So what we're gonna do and what I found that was super, super useful with my internet connection, okay, with my health class is that I muted everybody, and we just had questions and answers and things like that through the chat box, and that worked super well, and the system just worked. Okay, so that's what I'm gonna do right now. I'm gonna mute all of you guys. Okay, I will allow you to unmute yourself in case that you have an emergency, and I'm also going to want to have, for example, I have somebody here, which I don't know. Okay, Anais, I stopped your video as well so that we don't have any problems there, okay? Okay, so any questions, please just type in the chat box. This is how we're gonna be doing it. I'm also gonna download the chat box at the very end so that I can see how many people participated and what you said and things like that, okay? Okay, so we're gonna start with a quick analysis of biotechnology that is relevant for today. You guys hopefully already saw my my video, my screencast video of biotechnology, PCRs, electrophoresis, technology, cloning, all of that. That is still relevant today, but what's really revolutionizing biotech is CRISPR. So we're going to look at CRISPR in a little bit, and we're also going to look at COVID-19. So it's a virus. You guys already know what the virus is. It's called SARS-CoV-2, and it is what's causing our pandemic right now. And I want to analyze what type of biotech can be used to help us prevent or to help us cure COVID-19, and that it has to do a lot with the other assignment that you guys completed on March 9th, which was the COVID-19 questions. There was certain articles and questions about an article that was talking about how we can speed up the vaccine that's trying to be developed by scientists all over the world, and how vaccines are not just one thing. They can be developed in very different ways, okay? So we're gonna discuss that right now. Okay, so let me see if I can, okay, no, there, okay. Just give me one quick second, sorry. I wanna make sure that I've got this organized. Okay, so agenda, the warm-up is gonna be moved out to the closure, which is our quiz. We're gonna be discussing the answers to the COVID-19 biotech article that we were reading about speeding up the search for vaccines. We will be talking about something that's called RT-PCR, which are reverse transcription polymerase chain reactions. You guys should already know what the polymerase chain reaction is. Okay, polymerase keyword over there. Okay, so it has to do with a DNA polymerase, so it has to do with replicating. Okay, and we're gonna try to think if it could potentially be used. And we're gonna think about how it's actually used today to test whether you have SARS-CoV-2. So it is the biotechnology that is used in the test kits to see if you have this virus. And we're gonna review then why CRISPR is so important. So, okay, so let's first talk about what is a vaccine. So a vaccine can be any inactive part of the virus. Does it have to be the full virus? Please answer in the chat box if you know. People, answer in the chat box. Does it have to be the full virus? No. Okay, what could it be? Only inactivated pieces of it. Yes, very good, Juan Daniel. So it doesn't have to be the full virus. It can simply be a piece of the virus. It has to be something that we know triggers your immune response. Okay, why aren't we just creating a whole bunch of vaccines if we've got the SARS-CoV-2 virus? Why aren't we already just creating this vaccine? How do we traditionally, that's a better question. Requires replication of those parts, okay? How do we traditionally create viruses? Does anybody know? Sorry, how do we traditionally create vaccines? Okay, vectors, that's super good. That's actually how we're trying to do that today, but that's not actually how we originally created them. What we did originally is we would use a culture, a medium filled with cells, and we infect them with the virus. And then we allow them to replicate to the virus, okay? But it can be, you're not just replicating a little piece of the virus, you're actually making it so that those cells get sick, right? And they start replicating a whole bunch of virus, and then you just filter out the viruses that have been created from the host cells, and you've got the virus, and then you inactivate that virus to create a vaccine. So you're actually growing a virus. It's not like you are just producing or engineering a part of the virus. You grow the entire virus, and that takes a whole bunch of time. So traditionally, those vaccines take years to make because you need to be able to grow enough virus to inactivate enough virus to have enough vaccines for people. Okay, so that's also why we're not trying to think about how we would do this traditionally with SARS-CoV-2 because we don't have the time. So how can we actually find a way across growing the actual virus and doing this faster? There's different things that we can do with biotech. So the 3 different things that we can do, first of all, we can transform the virus RNA into DNA. Okay, this is an RNA virus. SARS-CoV-2 is an RNA virus. So we can simply transform that RNA into DNA through reverse transcriptase. Okay, remember that there's a enzyme that is called reverse transcriptase, and it's actually what HIV has. So what HIV already has in its DNA, and a way to code for this enzyme is it has its genetic material in the form of RNA. So it inserts it into its host. But instead of just producing that RNA continuously and making viral copies and then lysing the host, what they do is a lysogenic cycle. So they actually use that RNA, they use the enzyme that they produce themselves, which is reverse transcriptase, to create a DNA copy of that RNA, and then they insert that DNA copy in the host's chromosome. So in that way, they are forever embedded in that chromosome, and they're forever chronically in that person. That's why HIV is not curable, because of the fact that it is always, and it stays embedded in a person's chromosomes. Okay, so that's a chronic disease. So what you can do is you can use that information with reverse transcriptase, and you can transform the virus RNA into DNA, and then you insert it into a bacteria. So this is where we use what you're saying, Ana Belén. We use a vector. So we insert that into a bacteria through plasmids, and the bacteria can now produce a whole bunch of inactive versions of the virus. So you're not growing the virus itself. What you're growing is the code for what you're expressing is the code for the virus, and you can manufacture this code as well so that it can become an inactive version of the virus, and in that way you don't have to grow the virus. So it is a alternative to growing the virus, and since we know how to do this because we've done this with, for example, insulin in bacteria, this is called bacterial transformation, please remember that, then we can easily do this, and it is a bit faster. Okay, another way that we can get around this idea of growing the virus and creating vaccines from a culture of the virus is you can create mRNA sequences, messenger RNA sequences, of just the part of the virus that you think can activate the human immune system. So this has to do with what Juan Daniel was saying. It can just be some proteins of the virus. It doesn't have to be all the virus, and it's usually the spiky proteins on the outside, the corona proteins, that are the ones that are going to activate the immune system. So you find out what RNA sequence, what messenger RNA sequence is coding for these spiky proteins, and then you simply insert that mRNA sequence into our own cells. Because our cells, with our own translation mechanism, with our own ribosomes, will be able to translate that mRNA sequence into those proteins. And since now you've got the proteins that are foreign, that are identified as foreign, your immune system will get triggered and will start producing antibodies towards those proteins. And when you get the actual virus, your immune system will have the memory of how to produce those antibodies and will help you combat the disease much faster. Okay, you can also use something that's called a molecular clamp. Okay, so a molecular clamp, it's a clamp. If you don't know a clamp, it can be like una vincha. Okay, not vincha, what you use? Well, clamp. I don't know how to. Pinza. Thank you, Ana Belén. Una pinza. And what you do is that these spike proteins in the corona can actually change shape, and that is super bad for your immune system, because if it can change shape, that means that the antibodies that you're producing doesn't always target them. So what it can do, this molecular clamp, is it can attach to these spike proteins, these proteins on the outside of the virus, on the corona of the virus, and it can attach to them and prevent them from changing shape. And in that way, it allows your immune system to be much more efficient in finding these viruses. So those are 3 new types of ways that we're trying to develop And they are also called vaccines because of the fact that it helps your immune system combat the virus in much of a faster way. I would say that a molecular clamp is something a little bit different than a vaccine, but these two things right over here, the transformation of RNA into DNA and the creation of mRNA sequences, that's definitely an alternative to creating just an inactive version of the virus and inserting that into your cells. Okay. Besides vaccines, we can also think about a different approach to trying to, in this case, it's not a preventative approach, it's actually a treatment. The other cases, it can kind of be like preventative because you activate your immune system before you even come into contact with the virus. In this case, it's once you come into contact with the virus, you can enhance your immune system. How? Does anybody here know how an antidote works? Right, yes or no in the chat box. If you know how an antidote works. Maybe, I think. Okay. Okay, perfect. It's not actually very complicated. So an antidote is when you get a toxic or a toxin in your system, right? The poison of a certain animal, right? A toxin in your system. So there are antidotes to combat that toxin. And what they are is you use larger animals, larger mammals that have been in contact with that toxin. So let's say a horse. A horse that has been in contact with a certain poison will probably survive that poison simply because the horse is much larger than us. Okay, so something that might kill us might not kill a horse. So what happens is that poison goes into the horse system and the horse immediately starts producing antibodies against that toxin. Okay, so what can you do? You can take the antibodies from the plasma of the blood of the horse, you can filter them and everything, and then you can insert that into a human being. So when we're trying to combat a toxin that's trying to kill us, the reason why antidotes work is because we're pretty much just skipping a step. We're helping our immune system by inserting the antidotes that we know already combats that toxin because another animal has done that. So that's an antidote. We can use the same idea with COVID-19 and SARS-CoV-2. If a person has already been, they've already had the disease, okay? The virus, sorry. If a person has already had the virus, they've already started producing these antibodies, right? To be able to combat the disease. So what you can do is you can take the antibodies from, once the person, of course, has already been healed and everything, you can take the antibodies from the plasma of that person and you can inject it into a person that is combating the disease. And this is probably super good for what type of people? How old? I'll answer in a second, Mavic. So what do you think this is going to help? Who do you think this is going to help, this sort of technology? Older people or people that already have certain predispositions or diseases that doesn't allow their immune system to work very well. And who would you take the antibodies from? From young people, exactly. So Mavic, you were telling me, if you get the disease, can you get it again? The same exact strain of the disease, you can always get the virus, but if you have successfully combated the virus, that means your immune system now has memory of the antibodies that you're going to use to combat that virus. So theoretically, that's why we don't get the same flu 2 times. But the problem with viruses is that they mutate. So if a virus mutates in a specific region to change its spiky protein, then your antibodies that were already produced to attack that particular spiky protein don't work with a new strain. Okay, so yes, you could get the same type of coronavirus. You could get the similar strain of coronavirus, but it had to have mutated so that you can no longer attack it efficiently. If it is the same one, if it's the exact same one, you're not gonna get sick. No, you would simply be able to combat it super fast You kill the virus out of your system, and you're good. Could it also help babies that won't have their immune system totally developed? Yes. Yes, Kari, it could definitely help babies that don't have their immune system totally developed yet. The problem always comes with ethics, right? When we're using antidotes, we're using horses. In this case, we're trying to use other human beings. So I would say that there can be an ethical concern there. Okay. Okay. So let's move on. Okay. If you have any questions, please just continue writing them, and I'll stop and answer. Okay. So how can we actually test if a person has COVID-19? Okay. So remember that this is an RNA virus. So what we can do is we can transcribe it into DNA. Okay. So since we're doing the opposite, we're doing a reverse sort of situation, then we can use a reverse transcriptase. So this is called the reverse transcriptase PCR. Remember that whenever we're trying to check out if there is a certain sequence of DNA in anybody or admit. ¿Qué pasó? Sorry, Mavic left the session and then needed to be admitted again. I don't know why. Okay, so anytime that we need to check out or test if you've got a specific strand, a specific sequence of DNA in your genome, what you can do is you can use polymerase chain reaction. So what you're doing is that with particular DNA sequences, small DNA sequences that are primers that attach to the beginning of your sequence and to the end of your sequence, what you do is that you attach those primers there, and then you allow DNA polymerase, a DNA polymerase that you're inserting, of course, in those cells, to replicate those segments. And you do this over and over and over again, and that way you're cloning the segments. Okay, so at the end, once you've done all of these cloning situation and you have thousands of these segments, you can send this into a sequencing machine. And when you sequence it and it shows you that the sequence of that segment, you can test whether that is the sequence that you're looking for or whether that person does not contain that sequence. So in this case, what we've developed, so if you want to see it right over here, what we've developed are particular primers, particular DNA sequences that only attach to the sequence of the viral DNA of COVID-19, of SARS-CoV-2, okay? So once a person has SARS-CoV-2, those primers are going to attach to that DNA sequence and are going to start cloning it like crazy, okay? And the minute that the results come back, the doctor is going to be able to see that this person produced all of these sequences of COVID-19, so that confirms that the person is positive for COVID-19. If the person does not have the sequence for COVID-19, these primers have no place to attach because they can't find that sequence, so there will be no cloning of that DNA, and the person's negative for COVID-19. So that is reverse transcriptase. And the way, can you repeat that part, please? Branche, which part? Okay, so remember that when you're trying to create messenger RNA, all you do is you have a DNA sequence and you have a polymerase, an RNA polymerase, to create an RNA sequence from that DNA sequence. In this case, all you're doing is the reverse. You start off with an RNA sequence and you're going to create from that RNA sequence a DNA sequence. Okay, so what that is your reverse transcriptase that system, because transcriptase, normal transcriptase is from DNA to RNA. This is reverse because it's RNA to DNA. And the reason why we need it to be DNA is because you can only do polymerase chain reactions with DNA. Okay, hopefully that makes sense. Okay, and finally, CRISPR-Cas9 is CRISPR-Cas9. So hopefully you guys were able to watch both the Bozeman Science video and the TED Talk video. The TED Talk video is from the lady that developed this, and she is honestly a genius, and it is such a huge development. We've been able to use this sort of technology, very similar technology in the past. The problem with this and how we used it in the past is that it was never specific enough. So what we've been able to do in the past, what CRISPR actually does, is that it's able to cut the DNA strand, the double strand of any sort of genome, okay, in any specific place. And when it cuts that, remember that your own genome has a way to repair its DNA. It always wants to repair its DNA. So the minute that it gets cut, one of two things can happen. It can repair its DNA by simply rejoining the two areas that it was cut, okay? Or it can repair its DNA after you've added a specific segment that you want inserted. It can repair its DNA by joining the two ends into the area right next to the inserted sequence. So now you've inserted a whole new gene into a person's genome, okay? Now, why is CRISPR super innovative? We've been able to do this with restriction enzymes before, but the problem is restriction enzymes are not very specific. It is very hard to control where the restriction enzyme. Okay, so when the DNA is cut, it can either repair itself by attaching the ends that it was cut without inserting anything, or it can repair itself by inserting a genetic sequence that you have placed in the cell right in between the two strands that were cut. So now you have a full genetic sequence, a full gene, let's say, inserted right in the place where you cut that DNA. Okay, so it's kind of like this. You can just delete a gene, so you can cut an entire gene, and then the genome, is going to simply repair itself by attaching both ends of the cut. Or you can delete a gene and insert a new gene of interest, and the genome is going to repair itself by pasting or attaching, each end of the cut gene to the cut DNA to the new gene. In that way, inserting a new gene, okay? So why is this so innovative? Restriction enzymes have been used before to do this cut, but they're not specific enough. Why is this specific? It has right over here something that's called a guide RNA, and you can change this. What this basically does is that it attaches to the complementary DNA pairs in the genome. So if you want, you know exactly where to cut, for example, the beginning of a faulty gene in sickle cell anemia, okay? And you know what the code for the beginning of that faulty gene is, what you do is that you create an RNA version of that complementary DNA, and that's going to attach to that specific part. So you can cut in a very specific way, okay? So now once you have this guide RNA, what this guide RNA is attached to is called Cas9, and Cas9 is the enzyme that helps you cut, okay? So you cut the genome, you cut that specific section, and then what you can do as well is, as you're inserting CRISPR-Cas9, you can also insert the new sequence that you want instead of the sequence that you cut. And just based on your DNA way of repair, the DNA will insert that sequence into the place where it's cut. Okay, so let's think about how this could be helpful with COVID-19. Can we think about a treatment that could use, if this has anything to do, but in yesterday's class, exon skipping was similar to this. Yes, not necessarily, because you don't necessarily need to cut there. What you do is that from a immature RNA, you put a little segment of complementary base. Remember, RNA is single-stranded. So you put a little segment of complementary base, and what that does is that tells your spliceosome or your splicing machine that needs to be cut out. So what you're doing there more than anything, you're not altering the actual genome, you're altering the immature mRNA so that you can create a very different mature RNA and a different protein, okay? So again, to my question, can we use this with COVID-19? I'm gonna unmute you guys and let's see that we can wrap this up with just talking, okay? So can we use this with COVID-19? What do you guys think? Anybody, do you think we could maybe use this with COVID-19?
I think we can.
Yeah? How?
Yes.
Because we can cut like the section where the DNA of the
Okay,
virus
sure.
is inserted into our DNA, and that way, like, eliminate it.
Okay, let's remember that COVID-19 is not like HIV, right? It doesn't insert its DNA into our DNA. It doesn't actually have DNA, it's RNA.
It uses the cell to replicate itself.
Okay,
So
So you couldn't use the CRISPR-Cas9.
we
I
could?
think you couldn't. I think
Okay, why?
because if it doesn't
Because-
contain
yeah.
DNA, then we don't have any part to cut it out, so it wouldn't work.
Okay, cool. So that's
gonna-
kind of, yeah, that's kind of the idea. For this particular case, we can't. Can we use this with HIV?
Yes.
yes, because this embeds into your DNA, such as, for example, herpes or something.
Exactly. So you could potentially cure these chronic diseases, which are like HIV or herpes, which are inserting their DNA into your genome by simply deleting that
Cutting out
gene.
the gene.
Okay, why are we
Okay.
not using CRISPR?
Because
I
of
just
the
don't understand.
ethics of the people.
Because of the ethics of the
Mhm.
people, yes, because of the fact that we still do not know 100% all of the unintended consequences, okay? What happens if you're cutting a gene that has multiple functions? Yes, it might be a deleterious gene for a particular thing, but it might be keeping you alive for something else, okay? So that can be a problem. Isa, I think you had a question.
Yes, I was confused. Why can't we use the CRISPR-Cas9 in COVID?
COVID-19 is an RNA virus. Okay, so this is particularly made so that it can attach to DNA. And I would think that there could be, I would never say 100% no, because remember that CRISPR was actually discovered from bacteria. And it was a way that bacteria was able to splice and break viral genetic information so that they could not get infected. So I'm thinking that with a little bit of engineering, CRISPR could potentially be used. Just for right now, for the purposes that we have seen CRISPR being used, it's not necessarily the case.
And could CRISPR maybe be used like for detection of viruses, like for diagnosis?
Before you use CRISPR is more a gene editing tool. Before you use CRISPR for diagnosis, what you can simply use is a PCR.
Okay.
Because all you want to do is you want to clone as much of the reason. Why do we use PCRs, people? Why don't we just get a person's, for example, the cell of a person and just sequence that cell and see if that person has a particular disease? Why do we use PCRs in order to make sure that that person has a particular disease? What do PCRs do?
I'm not sure.
You're not sure? Okay, that's okay. When you have one cell, you have very little genetic information, right? Or when you have maybe 2 cells, 10 cells, 100 cells, you don't necessarily have a lot of genetic information. If you have, let's say, 100 cells, all you have is 100 copies of a particular gene that you're trying to figure out. So that you want to diagnose if somebody has cystic fibrosis. Cystic fibrosis is going to be a particular DNA segment that has a mutation and that you can detect, right? The problem is that when you only have a limited amount of cells, you have a limited amount of that mutation. You have a limited amount of that gene. So how can you amplify that gene so that when you sequence it, you can be 100% sure that that gene is there? You amplify it by using this PCR. And these PCR, what it's doing basically is just attaching a whole bunch of polymerases to that gene of interest and replicating it one time after another, after another, after another, after another. So now you don't have
Speeding it up, right?
100 copies of that gene, you've got 1 million copies of that gene. And then you send that to sequencing and it gives you a much more reliable result of what you actually have. Does that make sense?
Mhm.
Sophie,
Yes.
Yes.
this is what you mentioned yesterday that we needed to heat up a strand or what?
Yes. So PCRs work. Of course, you're trying to replicate a gene of interest that's within a genome. It's within a double helix of DNA, right? So the double helix is attached. So how can you make it unattached so that the polymerases can start replicating each segment of interest? You need to heat the DNA up. Whenever you're heating something up, the DNA of the double strand will break open. Okay, so you heat it up a whole bunch, and then the DNA polymerase is going to attach and start replicating the separated strands, and then you cool it a whole bunch, because like when you cool it, what you allow is that the strands that have just been replicated, they can join and they can become functional DNA, let's say. Does that make sense?
Yes.
Yes. Okay, so what we're going to do is for the next 10 minutes, you guys are going to be taking that mini 5-question quiz. And what I want to show you guys is what you're going to be doing for Monday. Monday, we're going to have that summative FRQ. So please have a paper and a pen ready. Okay, I'm going to post the FRQ online, but then you guys are going to be writing it down in a paper that you're going to send a picture of it to me.
Are we going to have a live session to take it?
Are we going to what?
Have like a live session when we take it?
We're going to have a live session when you take it. That's going to be around a 20-minute session. Okay? Okay, then, so that's going to be at 2 p.m. on Monday. Okay? Then you're going to complete this weekend. You're going to try to complete the AP Classroom personal progress checks. There are multiple choice and FRQ.
That's only for completion, right?
Yes, that's for completion. For the FRQ, people, question number 1, if you want to, question number 2, I'm thinking that you guys are going to be able to do it with no problem. Question number 1, if you want to work on that together, like have a little session where you're talking about this together, I'm allowing you to, because question number 1 is difficult. Okay? And then please remember that you're gonna have to complete the reading of chapter 23 for next Tuesday. Okay, that's also all on Schoology. It's all been posted on Schoology. So does everybody understand what you're doing and your plans for next week?
Yes.
Yes. Okay, so you guys can log out, and what you guys are going to be doing, remember, please, right now, before the class ends, you guys are going to be taking that quiz and click submit, and then I'll be grading that quiz. If you have technical issues, WhatsApp, please. I'll be checking my WhatsApp right now. Okay?
Do we have to turn on the lockdown?
Yes, you have to turn on the lockdown. That's the entire idea.
Coffee, when are we doing the collaboratively, like question 1 from the personal progress check from the effort?
That's up to you guys.
Ah, okay.
Yes, it's 100% up to you guys. All I'm saying is I'm allowing you to do it that way.
Okay.
Okay? Okay.
Sophie,
Okay, thank you guys.
Sophie, right now we take a quiz.
Yes, the AP Classroom Gene Regulation Quiz. It's 5 multiple choice questions. Okay?
Okay, bye, Sophie.
Okay, bye guys. Bye-bye.
Bye, Sophie.
Bye guys.
Bye, Sophie.
Bye. A ver, ¿a qué hora empieza mi otra clase? 9:40.